Good morning students. From today we are starting a new topic called as hemoglobin. In our syllabus, competency number 6.11 and 6.12 is allotted for this topic. So we will start with competency number 6.11 and today's topic is structure of hemoglobin. All of us know that hemoglobin is found in RBC. It has globular shape. If you remember the structure of proteins, there are two types of structures found in proteins, globular proteins and fibrous protein. So hemoglobin belongs to globular class of proteins. It has molecular weight 67,000. It is a conjugated protein because globin, which is a protein part, combines with non-protein part called heme. It has quaternary structure. Quaternary structure means when a protein is made up of two or more polypeptide chains. Here hemoglobin is made up of 4 polypeptide chains and each polypeptide chain along with its heme is called as subunit. Now 4 such subunits are found, therefore hemoglobin is having quaternary structure. So each hemoglobin molecule has 4 heme and 4 globin chains. Coming to the structure of hemoglobin, the basic structure of hemoglobin is made up of pyrrole ring. Heme is made up of 4 pyrrole rings. Each pyrrole ring is a heterocyclic ring. Each pyrrole ring has 2 sides. It is denoted by S in this figure, where 2 different groups are attached. These groups are called as substituent groups. Now the substituent groups present may be methyl group, propyl group, vinyl group and acetyl group. Now depending upon the presence of these substituent groups on these pyrrole rings, various types of porphyrin rings are formed that we will see later on. Now structure of porphyrin. 4 pyrrole rings joined together by methynyl bridge. Methynyl means CH bridges to form a tetrapyrrole ring structure called as porphyrin ring. Now this tetrapyrrole ring has property to absorb light. These pyrrole rings are numbered as first, second, third and fourth, and their bridges are named as alpha, beta, gamma and delta. For example, alpha bridge is present between first and second pyrrole ring, beta between second and third pyrrole ring, gamma between third and fourth pyrrole ring and delta between fourth and first pyrrole ring. Similarly, the substituent groups which are attached on each pyrrole ring, they are also numbered as 1, 2, 3, 4, 5, 6, 7, 8. For example, the substituent groups present on first pyrrole rings are 1 and 2. Substituent groups on second pyrrole ring are numbered as 3 and 4. Substituent groups on third pyrrole ring are numbered as 5 and 6. Substituent groups on pyrrole ring 4th are numbered as 7th and 8th. On the right side, I have shown the diagrammatic representation of porphyrin ring. As I have told you in the previous slides, depending upon the attachment of substituent groups on pyrrole ring or porphyrin ring, there are 15 different types of porphyrin isomers are found in nature. Out of these 15 porphyrin rings, only 5 types of porphyrin isomers are found in our body during the synthesis of heme, or they are intermediates during the heme synthesis. And these 5 types of porphyrins found in our body are uroporphyrin 1, uroporphyrin 3, coproporphyrin 1, coproporphyrin 3 and protoporphyrin 3. Now out of these 5, the last protoporphyrin 3, which is the 9th isomer, is found in our hemoglobin molecule, whereas upper 4, they are found in the as a intermediate during heme synthesis. When we study heme synthesis, at that time you will understood this better. So here now just remember, protoporphyrin 3 is the porphyrin found in normal hemoglobin molecule out of these 5. So depending upon the attachment of substituent groups on porphyrin ring, these are the different types of porphyrins. First is uroporphyrin 1. Here the substituent groups are acetyl group and propionyl group in an alternating manner. So at 1, 3, 5 and 7 position you have acetyl group, and even on even number, that is 2, 4, 6, 8, you have propionyl group. In uroporphyrin 3 you will find the same substituent groups, that is acetyl and propyl group, but there is alteration in the position at 7th and 8th position. In uroporphyrin 3 you have propyl group on 7th position and acetyl group on 8th position. Next porphyrin is coproporphyrin 1. In coproporphyrin 1 the substituent groups are methyl group and propyl group, again in a symmetrical manner, that is at odd position, that is 1, 3, 5 and 7 you will have methyl group, whereas at even position, that is 2, 4, 6, 8 you will have propyl group. In case of coproporphyrin 3 the substituent groups are same, but there is again alteration at 7th and 8th position. On 7th position you will found propyl group, and on 8th position you will found methyl group. Now if you see here, porphyrin 1 have symmetrical arrangement of substituent groups, whereas porphyrin 3 have asymmetrical arrangement of substituent groups. The last porphyrin is protoporphyrin 3, which is, as I have told you, found in our normal hemoglobin molecule. It has 3 substituent groups: methyl, vinyl and propyl. Here you will see the arrangement. Methyl group is present on all pyrrole ring, but vinyl group is present on first and second pyrrole ring, whereas propyl group is present on third and fourth pyrrole ring. Now out of these 3 substituent groups, that is methyl, vinyl and propyl in protoporphyrin 3, only propyl group is hydrophilic, whereas the methyl and vinyl groups are hydrophobic in nature. So this point is important to remember for our future hemoglobin structure. As I have told you, series 1 porphyrins are symmetrical distribution of substituent groups. Series 3 porphyrins are asymmetrical distribution of substituent groups. In our biological system, that means in our body, type 3 porphyrins are predominant. After completing the structure of porphyrin, we see the structure of heme. In a porphyrin structure, when iron is introduced, it is called as heme. So this iron is attached to the 4 nitrogen atoms of a porphyrin ring by 4 coordinate bonds. This is a normal heme. Again you see it is this ring is protoporphyrin 3 ring. Here methyl and vinyl groups are hydrophobic, whereas propionic group is hydrophilic. Now about the iron in heme. In a heme, iron is present in 2 forms, ferrous form and ferric form. Ferrous form is hexavalent, whereas ferric form is pentavalent. That means ferrous form forms 6 bonds, whereas ferric form of iron forms 5 bonds. Ferrous form of iron binds with oxygen. Ferric form of iron does not bind with oxygen. Ferrous form of iron is found in oxyhemoglobin and deoxyhemoglobin, whereas ferric form of iron is found in methemoglobin and hemoglobin M. Methemoglobin and hemoglobin M are different. Now, how this ferrous and ferric is converted into ferric form? Ferrous form of iron, when loses one electron, it get converted into ferric form. Remember, this ferric form of iron does not carry oxygen, which is the main function of hemoglobin. But ferric form of iron is more stable form of iron. Therefore, iron always have tendency to convert into ferric form. Whenever the iron present in a hemoglobin molecule exposes to hydrophilic or aqueous environment, it readily loses the electron and get converted into ferric form. Functions of heme. I am not talking about hemoglobin, I am talking only about heme. It binds with oxygen molecule for transport, and just I have told you that ferrous form of iron binds with the oxygen molecule and transport the oxygen molecule from lungs to other parts of the body. Secondly, heme absorbs light and gives red color to the hemoglobin. I have already told you that porphyrins have tendency to absorb light. So in heme, protoporphyrin 3 ring structure is present, so it absorbs light and gives red color to the heme, or red color to the hemoglobin in our body. Now, after completion of heme, we will see structure of globin chains. Globin chains are polypeptide chains made up of different amino acids. 4 different types of globin chains are alpha chain, beta chain, gamma chain and delta chain. Alpha chain is made up of 141 amino acids, whereas beta chain is made up of 146 amino acids. Both alpha chain and beta chain have proximal histidine and distal histidine residue. Now, what is proximal and distal histidine? Now, in this structure, I have shown you centrally heme and peripherally a globin chain. Now, this globin chain contains many amino acids. Just we have studied 141 amino acid in alpha chain and 146 amino acid in beta chain. Now, out of these amino acids, few histidine amino acids are present in the globin chain. Histidine is a amino acid. Now, 2 histidine molecules are important here. They are called as proximal histidine and distal histidine. Iron is directly attached to one of the histidine nitrogen and forms a coordinate bond. That histidine is called as proximal histidine. In another case, when iron is in ferrous form, then it forms a coordinate bond with oxygen molecule, and that oxygen molecule will form a hydrogen bond with the another histidine molecule, which is called as distal histidine. It is not mean that only 2 histidine molecules are present in the globin chain. Actually, many histidine molecules are present in a globin chain, but these 2 histidine molecules forms direct or indirect bond with iron atom. Therefore, they are separately named. Now, proximal histidine means it is nearer to central iron atom, whereas distal histidine means it is little bit far away from the central iron atom. Therefore, it is named as distal histidine. Now, in alpha chain, 50th position distal histidine is present, and at 87th position proximal histidine is present. Now, this 50th and 87th position means they are numbered in a primary structure. When we unfold the globin chain, and if we try to number the amino acid sequence 1, 2, 3, 4, at that time distal histidine will get 58th position and proximal histidine get 87th position in a straight chain structure. But when they form a hemoglobin structure, there is folding in the globin chain, and during that folding 87th amino acid of that is histidine, 87th histidine residue come closer to iron, and hence it is called as proximal histidine, and 50th histidine molecule is placed little bit behind in the folding. So similarly in beta chain, 63rd histidine is called as distal histidine, 92nd histidine is called as proximal histidine. Gamma and delta chain are similar to beta chain, that means they both possess 146 amino acids and the position of proximal and distal histidine is same. What is different? But they have different amino acid sequence as compared to beta chain. Therefore, they are named as gamma and delta. So, this is the structure of one hemoglobin subunit. Now, how this heme and globin combines? Heme and globin combines by different bonds. Just I have told you, iron, which is centrally placed in a heme structure, forms direct coordinate bond with the nitrogen atom of imidazole group of histidine, that is proximal histidine. And if the iron is in ferrous form, it forms a bond with oxygen, and that oxygen forms a bond with distal histidine, nitrogen. So these are the two attachments. But if iron is in ferric form, this bond does not exist. The bond with distal histidine does not exist. Apart from these two bonds, the propionyl group which are present on 3rd and 4th pyrrole rings. I have told you that propionyl is a hydrophilic group. It loses the hydrogen atom and get H plus ion and get negative charge. Now this negative charge forms a electrostatic bonds with a basic amino acid present in a globin chain which has positive charge. All of us know that basic amino acids have positive charge, acidic amino acids have, acidic groups have negative charge. So propionyl group is an acidic group, whereas arginine is a basic amino acid which has positive charge. So there is a two electrostatic bonds are present between propionyl group of 3rd and 4th pyrrole ring and two arginine amino acids present in the globin chain. So this is the attachment between heme and globin. In such a way, hemoglobin subunits form. Each globule chain has helical structure. Alpha chain have 7 helices and beta chain have 8 helices. These helices are named as A, B, C, D, E, F, G, H, etc. Now heme is present in between E and F helices. Now this heme is protected by the globin from the aqueous environment which is present outside. Heme is a hydrophobic compound, so it should be protected from aqueous environment. So heme get incorporated into the helices of globin chain, and then globin chain protect the heme from the aqueous environment. Now functions of globin chain: it protects the heme from hydrophobic pockets, from exposure to hydrophilic environment, and thus maintain the iron in ferrous form. I have told you that iron is in two forms, ferrous form and ferric form. ferric form is more stable form. So iron always try to achieve a ferric state which is more stable. So whenever there is a exposure of heme with hydrophilic environment, the iron get converted into ferric form, and this ferric form of iron does not carry any oxygen. So globin chain protect the heme from aqueous environment. And second function of globin chain is it binds with the carbon dioxide molecule and help in the transport of carbon dioxide. That we will see in detail when we study transport of gases. This is the structure of hemoglobin subunit that I have already explained. These are the attachments between heme and globin that also I have explained. Propionic acids of the porphyrin ring form electrostatic bonds with two separate arginine residues of globin chain. Iron forms one coordinate bond with nitrogen atom of imidazole group of proximal histidine. When iron is in ferrous form, it forms one more coordinate bond with oxygen, which forms hydrogen bond with the nitrogen atom of imidazole group of proximal histidine molecule. Now this is the complete structure of hemoglobin molecule. 4 hemoglobin subunits combines and forms one hemoglobin molecule. Hydrophobic bonds, van der Waals forces and some electrostatic bonds are present between these 4 hemoglobin subunits. So this is the structure of complete hemoglobin molecule. So if you see, it has 4 heme subunits and 4 globin chains. This is the diagrammatic representation of the hemoglobin molecule, which shows 2 alpha chain and 2 beta chain. I have told you in the previous structure, 4 subunits binds together by hydrophobic bond, van der Waals forces and some electrostatic bonds. Apart from this, when hemoglobin is in deoxy state, these 4 subunits are bind together by salt bridges. So these are the salt bridges, but these salt bridges are not present in oxyhemoglobin. They are present only in deoxyhemoglobin. Now if you compare one subunit, or it is made up of heme and globin. If you compare the weight of heme and globin, the 4% of the weight of hemoglobin is made up of heme, whereas 96% of the mass of hemoglobin is made up of globin chain. Normal hemoglobin in the blood is 14 to 16 gram per deciliter, and in females it is 13 to 15 gram per deciliter. Normal adult blood contains 97% of hemoglobin A. A stands for adult, 2% hemoglobin A2 and 1% hemoglobin F. F stands for fetal hemoglobin. Hemoglobin A, that is normal, it forms 97%. It is made up of 2 alpha chain and 2 beta chain. Hemoglobin F, which forms 1%, made up of 2 alpha and 2 gamma chains. Hemoglobin A2, which is 2%, made up of 2 alpha and 2 delta chains. In all these hemoglobin molecule, heme structure is normal, only there is a difference in the globin chain. All these are normal hemoglobins. So this is about the structure of hemoglobin. Thank you very much.